In this tutorial, we are going to analyze breast cancer RNA sequencing data from the Cancer Genome Atlas project. To begin, we're going to first download the RNA-Seq data from a website called cBioportal. I have cBioportal open already, and under cBioportal, if you go to Datasets, and then there will be a list of different cancer data sets here. You can download one of these. In this case, I'm going to download the 2015 invasive breast cancer data set, which can be found right here. So if we click this button, it will download a zip file. It will extract it. And within there is an RNA-Seq expression file. We're going to use the non-z-score normalized file because the z-score normalized file has NAs in it, which we'd have to replace with some of their values. So after we've downloaded that, in addition to the expression data, there's also a clinical file. In this case, we're going to go to the ER status column within that clinical file, and we're going to extract out the sample ID, the TCGA sample ID, and whether the ER status is positive or negative. I've already done this. I've already actually created these files and pre-filtered them. And in case of this expression file, the only way that we filtered it is we filtered it to the top 2,500 covariating genes. And so we used a coefficient of variation, which is the standard deviation divided by the mean, as a sorting parameter. We've also filtered for genes that only are expressed in 100 out of the 800 or so samples. And we have also added an additional filter to that query to require that the expression value is above a value of 100. I believe it's TPM in this file for those 100 samples. So we've already done that filtering. That filtering was done in Excel. We also created this groups file. The groups file here has only 3 columns. The first column is the sample ID. The next column is a number for the different groups. The group here is shown on the right. In the third column, negative or positive. This is ER status. You can see that ER positive is going to be 2. And the expression file, as I described it, is just a simple expression file that has a first column is the gene symbols, the UGO gene symbol, and the first row is the TCGA symbols. And these are the non-log expression values. It's fine to use log or non-log expression values to begin with, which are scaled. Okay, so we're ready to begin. We can open our version of AltAnalyze. If you haven't installed AltAnalyze already, you're going to need to download it from the AltAnalyze website. In this case, I have a fresh version here. You would double-click on AltAnalyze if that works for you. If this doesn't work for you, see the previous video, which describes what to do on a Mac. If that does not work for you, it's a common problem on some systems. You say begin analysis, continue, and it's asked you to download a database. You would say continue. And you would also, at this point, say which species you want to download. Please note the Ensemble version is linked to a specific genome version, so you'll need to go to the Ensemble website and see what genome version is supporting this analysis. This is a text file in this case, so it doesn't really matter. Here we would select Homo sapiens and say continue. Once this database has downloaded, you'll be returned to the main menu. From the main menu, you'll be able to select what additional parameters or analysis options you want to use before you perform either an automated analysis or visualize your data in various ways. In this case, we're going to perform a number of data visualizations first, and so we select Continue. And under this second menu, we're going to select Additional Analyses. Under Additional Analyses, we're going to select Dimensionality Reduction. And here there are a number of options to select from. First, we're going to select an expression file. We're going to select this expression file here. And we're going to perform principal component analysis. To do this, the default algorithm is listed as UMAP. We're going to change this to SVD. And we're going to change this z-score normalization to yes. And now we're going to perform a proper principal component analysis. If we do this and select continue, this analysis typically only takes a few seconds or less than a minute in almost all cases. And you get this 3-dimensional plot by default. And you actually can select this plot and you can spin it around. And you can actually see what variance is associated with these three principal components. The reason there are two colors here, blue and red, is before we began, we created this groups file. This groups file has the identical name as the expression file, except we've swapped out groups for expression file. And so the program will automatically recognize this groups file as having annotations, which it should layer onto this variance-based map. And you can see in principal component 1, PC1 is actually principally defined by this difference between ER positive and ER negative status in terms of the clustering of the samples, which you really don't see clearly within PC2 or PC3. So this is useful for visualizing the data and seeing how different principal components associate with different annotations that you may have. But once this is saved, there's this folder within the input folder that's called data plots. Data plots now has two additional files in it, the PDF for this PCA plot, the PNG for that plot, and this folder PCA, which contains this expression, this file, which you can open up in Excel or text editor, and you can see it lists the top genes correlated with principal component 1 through 4 and anti-correlated through principal component 1 through 4. So you can see specifically which genes are associated with ER positive versus ER negative status, and not surprisingly, the top correlated gene is ESR1. So this is cool. This reproduced a lot of prior known biology in a very simple analysis without really instructing the program to know anything about ER positive versus ER negative status. So there's a number of things you can do within this PCA or this dimensionality reduction view that we're now going to explore. So we can actually go back to this view and select some of these different options. The first option is if you want to display the sample names next to each dot. You don't want to do that here because there's too many dots. You can choose to perform, choose to use a different algorithm or perform to do two-dimensional versus three-dimensional reduction PCA analysis. If you select 2D, the plot will look very similar to what we first saw, but you'll just see now PC2 and PC1 side by side. And again, that same plot will be generated in PDFs for the analysis. We can also choose to make a UMAP plot, and so we can change this to UMAP or TSNE. In this case, we'll use UMAP. The first time you generate a UMAP plot, it takes a little bit longer than the next time, and the reason for that is it's basically producing coordinates and it's saving those coordinates, and those coordinates are stored here in the scores file, and it reuses those in subsequent analyses, so basically the analysis time is much faster. And again, you see that there's a difference between the positive and negative samples. They're basically dividing out on this UMAP X set of coordinates. In addition to simply just viewing the data within a PC plot or a UMAP plot, you can also color genes within this plot. And to do that, again, we could look at, I think the top gene associated with PC1 was ESR1. And so you can type in ESR1, and we can see how the expression of ESR1 differs among these different populations. And you can see that ESR1 is expressed highly in the positive population we saw here on the left and lowly in this population on the right. And so this is a very useful way to look at the expression of one or more genes. We can actually go back and look at the anti-correlated genes for PC1. So we'll go ahead and do that. And the top anti-correlated gene was MAMLD1. So we'll go ahead and we'll take that. And we'll add that gene here. And so now when you view these two genes, it's actually going to color by the where the predominant expression of these two genes are. And you can see that MAMLD1 is now predominating within the estrogen receptor negative, and ESR1 is predominating the estrogen receptor positive. And so these are useful ways, again, to compare and contrast different genes in terms of their predominant expression within a plot. When you have a PCA analysis, and we're going to switch back to SVD, And we're going to actually, in this case, say we're going to store the top 200 component genes as. And so we're going to call this BRCA, PCA, and I've done this before, so later on we're going to see similar names to this when we select this gene set later, but we're storing basically those top PCA loading genes that we were seeing in that text file in a parameter within the database of AltAnalyze. So we do this, and you're not going to see anything special at this point. You're just going to get that PCA plot that we previously saw, but now it says exporting gene set to stored gene sets, and so we can access that later. So now if we go to hierarchical clustering, it by default is loading the same expression file. I'm going to go ahead and hide this data plots folder. And there's a number of default options for clustering here. And so I could cluster all 2,500 genes, and that's fine. That's a reasonable thing to do, but often there's a number, we may actually want to select fewer genes that are the most variable. And so in this case, if we go to select gene set ontology to filter on, there's a number of different pathway and gene set categories here. I have some custom ones in here, but there's also stored gene sets. And here under stored gene sets, I'm going to select the option that I just created, which is this BRCA PCA. And if I now select, say, continue, we're going to generate a heat map from those original 2,500 samples or genes, but now filtering for the 714 genes that were associated with those first 4 principal component analyses, or principal components. And you get a heat map that looks like this. It's an interactive heat map in the sense that you can, if there are features in this heat map, such as these genes, you can click on them, and it will open up a web viewer, and you can actually see information on this gene from gene cards. You can also click this open heat map in tree view, and if you have Java installed properly, sometimes this requires some finagling to get Java working with this little program here called tree view. You can now interact with the components of this heat map, and you can see which genes are associated with different subclusters here that you may want to use for further analyses. So this is a very basic heat map view of the data. The heat map viewer just takes a simple tab-delimited text file expression matrix like the one I showed you before. Again, you actually now see which cell samples are ER positive versus ER negative, and this is actually doing a decent job of clustering these samples with this default clustering metric. There are other clustering metrics we can do. If you've previously installed R on your computer, you can actually select HOPAC clustering. And HOPAC is a sophisticated method within R for clustering that's a bit more rigorous than standard hierarchical clustering methods. So you can exploit that. You can choose to actually turn off clustering for the rows or columns if you want. You can change how you want to view the expression data, row, median normalization versus mean versus no normalization, and you can choose not to use these stored gene sets or other gene sets to filter on. But importantly, you can also perform a pathway or gene set enrichment analysis directly in the plot. And so we're going to go ahead and do that. In this case, I'm going to choose, I think, pathway commons, which I believe actually was, I lost it here. There's too many categories here. Let's see, and they're not in alphabetical order. So pathway commons is, it's somewhere here. I apologize that I'm not seeing it. Oh, there it is, pathway commons. Okay, so I'm going to select, say, continue. And this time it's going to take a bit longer to do the clustering because it's going to connect to R. And it's going to use this more rigorous, sophisticated clustering method called HOPAC. If you wanted to run this, you would first have to download R for the computer, install it. The program does try to install HOPAC and bioconductor, which are the 2 dependencies automatically. But on different Mac operating systems, there may be the gatekeeper may prevent that from AltAnalyze installing those things. So you may need to install both bioconductor and HOPAC to have this run. After the clustering is performed, and you'll see this HOPAC output written, and it does this for both the columns and rows separately when it's done, it's now going to perform this gene set enrichment analysis. And the gene set enrichment analysis is performed using a tool called GoLead that you see here. And you can see the status of this different tool if you click on here. If it's not running and really taking up a lot of CPU time, it will eventually go down. But it'll tell you the status of GoLead. GoLead is basically performing a number of statistical tests to look for enrichment of each of the gene clusters that are produced by HOPAC. And those are going to be the clusters on the left. And it's going to be displaying those terms on the left side of the heat map. And those terms are actually going to be interactive in the sense that you can actually click on them and view what genes are associated with each. So in this case, the analysis is done. I'm going to expand this heat map a bit more. You can see again that the ER positive samples are listed on the right, the ER negative samples, which are the poor prognosis, are listed on the left, and you can actually see these different gene set categories that are shown on the left here now. And if you click on one of these, so let's say cell cycle mitotic, we're going to see a network, and the network is going to show the different gene set terms that were associated, that were enriched within that particular gene cluster, and it's going to show the genes that are connected to those different terms on the left. And as you can see, some of these genes are central in the consensus that they basically connect to multiple terms here and are basically core genes within that within the gene set context within that cluster. So for example, BRC5 is one of them. So you can do that for any of these terms. You see there's additional gene markers on the right now. That's because we have additional clusters that HOPAC has identified. Again, it's a more rigorous clustering algorithm. And again, we could explore that by clicking on this tree view option here. So there's a number of other things you can do within this heat map viewer. You can parameterize the filtering of these genes and so on and so forth, and that's covered in depth in another video related to just using this heat map viewer. But now we're going to close this program, or this window, and we're going to go back to the main menu, and we're now actually going to perform a differential expression analysis on the original expression file and groups file that we have. I should note that the GoLead results are also saved here in this window that popped up in this pruned results z-score elite file, and so you can always go back, open this file in Excel, and you can see exactly which gene sets were enriched in which particular clusters, and what are the associated genes with each of those gene sets along with associated statistics. So going back to the main menu and then to continue and then to process expression file, we're now going to select continue. For selecting the expression file, we're going to say select file, and again select the same expression file we selected before. We're going to select the output directory as this home directory. And when we hit continue here, you'll see a new folder is created here. The program automatically moves the expression file and the groups file here to this new folder called expression input, which is a folder that the program automatically recognizes, that it's now knows that all the core files were within this directory. So it's an important folder for the program. You're now going to be presented with this parameters window. And the parameters here are whether basically or not to perform different visualization analyses. In this case, we're actually going to say no for all of these at first. And the reason why is we just want this to be really fast. Each of the plots takes a little bit of time to generate. So we're just going to focus on the tabular results. If we select continue here now, we're presented with another window where we have the option to select different stringencies for our statistical analysis. In this case, we're going to say we want to restrict our analysis to adjusted p-value and FDR corrected, Benjamini-Hochberg corrected p-value for filtering. And there's a number of gene sets it can use for doing pathway enrichment analyses. Here, we're going to just say, we're going to look at these pathways because it's quick, again, for the purpose of this tutorial. Because we had already created this groups file, all these 800 samples are going to auto-populate with whether they are ER negative or positive. Select continue. We can now say which our comparison is. So we're going to say, actually, we're going to compare negative to positive and continue. and you'll see this dialog window pop up, which will show the progress of these different statistical analyses. Again, we chose not to make any of the nice QC plots or heat map plots and PCA plots the program can automatically produce, but that's something you can easily do just by saying yes to those different options. And many of those options are very useful, and I do recommend you use them. However, if you want to be very quick with the analysis like we are here, you would try to replicate the different options that we selected here. And so the analysis is basically almost done at this point. It's performed the differential expression analysis between the ER negative and ER positive patients. And now it's just performing this gene set enrichment analysis, looking at, it said the category is pathways, but it happens to be wiki pathways. And when it's complete, you'll see this dialog here that analysis is complete. You select OK. In this case, because we did not include those additional visualization analyses, there's no links to those results. However, if those results are generated, they are in these output directories here, and I can show you that in a moment. So I'm going to say close view and quit. And we're just going to go ahead and look at these different directories. And you'll see there's a number of new directories that have been created. There's PDFs here that describe what are the files within in folders within each one of these directories, if you're curious. So those are tantamount to readmes. And in this case, we're going to look into expression output, and we're going to focus on this one file called dataset TCGA. And so the dataset file here, I'm opening in Excel by right-clicking on it. This is usually not recommended, because if you do this and you now sort by gene symbol, you'll see that the genes at the top are actually gene symbols that are originally called sept1, sept3, sept4, for example, and their gene symbols have now been replaced by these dates, which Excel does automatically, and you can get around that by opening the file from Excel and choosing to import this column or these columns as text rather than as automatically determined fields. In this case, however, we're going to resort this file, and there's a number of statistics and annotations in this file that we would want to look at. First, we want to sort by the adjusted negative versus positive group, adjusted P. So this is the FDR corrected p-value. This is in this data set. This was a empirical Bayes moderated t-test, basically using a method is used in lemma in R. AltAnalyze replicates that method. And so here now we're looking at the most significantly differentially expressed genes with an adjusted p-value using this FDR cutoff. And we can see over here what the fold changes for each of these genes are. And then on the left, we can see what the names of those genes are, for example. So in this case, ESR1 was the most top significant gene, which is not a surprise given our principal component analysis. And we can see the pathways that ESR1 is associated with. So here you see various gene ontology process terms, see gene ontology function terms. So this is associated with DNA binding, beta-catenin binding, chromatin binding, transcriptional regulation. You can see where the protein tends to be located within the cell in terms of various compartments, and you can see the wiki pathways that this is also associated with, including estrogen receptor pathway. And so you could go to this wiki pathway here on the web, and you could see how these different genes are associated with that pathway and where they sit within that topological network. There's also a number of general categories shown here, including is the protein transmembrane or extracellular. It says this is protein's transmembrane. It tells you its putative microRNA targets are that can regulate repress this transcript from being translated. You see that it's a class as a transcriptional regulator and a receptor in its protein encoding. So this is all very useful. It also gives you the ensemble gene ID here, and you have the log2 expression values associated with the negative and the positive averages here for those two groups. You can see some genes are downregulated, some genes are going to be upregulated here. And so that really gives you a primer on how you can use AltAnalyze for differential expression analysis. You're probably going to have a number of different questions. There are a number of different outputs here, including the gene set enrichment analysis results, which can be found in GoElite. This is separated out for up and down regulated genes. In regulated, for example, there's additional nice little outputs you have access to, including putative gene regulatory networks. Here you see actually ESR1 is downregulated, negative versus positive. And you actually see it's indicated as blue. These red arrows indicate putative transcriptional regulatory networks. These are basically ChIP-seq-based predicted interactions, or from regulatory interactions from other databases. Its targets are also downregulated, and you can see other nodes in this network, including genes associated with cell cycle. So there's a lot you can get of AltAnalyze. This is really just a small taste. But if you have questions, contact us. We're happy to respond and walk you through how to optimize parameters or perform different analyses. So thank you very much.